This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1217, recorded on the 9th of May, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it's been a pluviose week, and we're continuing that trend today. Yes. It is pouring rain, little misty and foggy, 55 Fahrenheit, 13 C. Supposed to continue like this through halfway through tomorrow and then clear up and be lovely on Sunday.
Pluviose. I'd already forgotten that word.
Yes,
I didn't use it.
I looked it up, but I forget what it means.
Rainy.
Rainy. No
pluviose here all week. Pluviose here all week.
Yeah.
Wow. Okay.
Also joining us from Austin, Texas, Rich Condit.
Oh, yeah, I jumped the gun, didn't I?
That's
I
all
haven't
right.
been introduced.
It doesn't matter.
It's all right.
It's just so people
hi,
know
everybody.
it's just so people know who we are.
Yeah,
really.
I know. Okay, so it's here in Austin, Texas, it is 81 degrees Fahrenheit, 27 C. There's a kind of a high overcast, though Dixon is trying to creep in over my shoulder there, so
Yeah.
it's a bright overcast. We have had quite a bit of rain in the last few days, which is good because our main reservoir is down to record lows, and it's going to take a deluge to fill that sucker up. And I don't see a deluge in our future, but every little bit helps. I'm happy about that. So we're good.
Here in Chelsea, it's 15 C and rainy, cloudy.
Pretty similar to us then. Yeah.
You get a sip of water here.
Yeah, we also had we had a really dry spring until now, and now we're catching up. So similar situation to Rich. Everything was low, and now we got flood warnings.
I'm also under the influence of some sort of respiratory pathogen. Everything's going to be okay. And I actually did a check, and it is not SARS-CoV-2. It's just the usual thing. I'm providing a home for a little piece of probably RNA. Could be DNA. Who knows?
Eventually, we're going to have a little test swab that won't just tell you if you have SARS-CoV-2. It'll be, a whole panel.
Yeah, a whole panel.
A whole respiratory panel. That would be great.
Right.
So we had a letter to Clinical Update to Daniel. This guy said, I got COVID. I tested. I was positive. I got better. And then I started coughing again and sniffling. And it's been going on for weeks. And I'm negative for COVID. What's going on? Do I have long COVID? Do I have this?
No, you got something else, dude.
We said we got something else. And the next week he wrote in, it's metapneumovirus.
Ah, cool.
So he actually got tested. How about that?
Yeah, if you go to the docs now, they'll run a PCR panel on you that's got I don't know, 15 or so viruses and several bacteria. It's amazing.
Yeah, the test does
BioFire.
exist. It's just not we don't have an at-home antigen test for all this stuff.
Yeah, it's BioFire, the company, and they have these respiratory and gastrointestinal and neurological panels, but it's a PCR test, multiplexed.
I want one.
So it's not a consumer product, but you can get the data. You can go onto their website and they pump all the data up, anonymized, of course, and you can see trends over different seasons.
That's cool.
Right?
Nice.
Very cool.
That sounds like a pick of the week.
Yes, it really should be. I was in Emory a couple of years ago, and I was sitting at breakfast with my host, and this guy comes over. He said, "Are you Vincent Racaniello? I heard your voice."
I love that.
Said, yeah. So he sat down and he started talking. It turned out he worked for BioFire. He listens to TWiV, and he told me all about this website where you can get the data. I said, I thought you might like to know this. I like that when they recognize your voice. Right?
Yeah, that's really cool. It's happened to me a couple of times.
Well, if you listen a lot, then you're going to recognize people's voices. Otherwise,
I always like it when I contact a source to interview them, and they're like, "Yeah, of course I know who you are. I listen to TWiV."
Yeah, it's good.
Yeah, all right.
It's good. Well, if you like these programs, all of the podcasts, not just TWiV. We'd love to have your support. We are a nonprofit microbe TV that is the production company basically for these podcasts. We record them, we produce them, and it all is associated with costs. And we'd love your help in defraying those costs because we do not do ads. We have no source of income except you and other donations. We have a few foundations that donate, but not enough to run this. The running depends on you, our listeners. So if you go to microbe.tv/contribute, you will find a variety of ways in which you can contribute. We thank you for your support of science communication in these troubling times when science is being attacked by non-scientists who know nothing. Zero. And one of the papers we're going to do today is going to emphasize how much they do not know. Oh my gosh. Actually,
and not
Actually,
how
both
much
of
they
them,
don't not how much they don't know in a good scientific way, like, we do a
Yeah.
paper and we
Yeah.
say, oh, so many cool questions to ask, but how much they don't know in terms of how, like, they're 180 degrees wrong in what they do say.
Yes. And that the evidence that they say is evidence is not evidence
Yeah.
whatsoever. A couple of news items.
On that note,
Okay. So Vinayaka Prasad has replaced Peter Marks as the CBER director, Center for
biological evaluation and
Research. right.
research. Yeah.
This is a disaster. He's a critic of drug industry and COVID-19 measures. He's going to lead this is basically the division of FDA that does, among other things, vaccines. Okay.
This
And-
is a CBER. I think we've talked about this before. It's an unusual government, fundamentally on paper regulatory agency, but it's more than that because it's one of the, I think maybe the only that has active research laboratories going on site. Okay.
Yes.
And
And you know who works there, right?
Yeah, I know who works there, and I know a few others who have. I've been there.
Yeah.
There
There are a number of regulatory agencies that have active research
Okay.
programs, and also have their regulatory role. I mean, the one that comes to mind is the USDA.
Of
which
course.
is
Yeah.
a big operation that way. But CBER is the biologics part of FDA. They approve biologic drugs, which includes vaccines, but it also includes everything, all the protein-based stuff that's made in genetically modified organisms these days, which is, everything from monoclonal antibodies to insulin. And then there's the drugs division and the device division. So this is biologics, which is a major regulatory arm.
Yeah, I think it's interesting to point out that insulin is a GMO,
Yes,
right?
Mm-hmm.
it is
about
now.
that.
I mean, it was what was it previously? I think it was previously isolated from pigs or
Yeah,
something.
pig pancreas, and it takes a lot of pig pancreas
Yeah.
to make an infinitesimally small amount of insulin in a very laborious process.
Whereas if you grow it up in a fermenter now,
Yeah.
So there's an article in U.S. News I want to make a few quotes from. First of all, they say Prasad is the latest in a series of medical contrarians who's joined the federal government.
such a mild way of putting it.
He replaces Peter Marks, and Prasad, in a social media post, said Prasad likened Marks to a bobblehead doll that just stamps approval. This isn't true at all. He didn't do that. And it's really unfair to say that. It's a lie, basically.
Yeah.
So there you go. This is what we're going to expect from Prasad. Prasad is an oncologist, by the way. And I forget where he is. UCSF. He's a professor there.
His credentials on paper look great, but as we are learning, that doesn't necessarily.
There are quite a few people who fall into that category where you look at him on paper and you say, "Oh, wow, guy's got a medical degree, he must know what he's talking about," and but no, that doesn't guarantee anything.
Well, when COVID came, he decided that he was a COVID expert, and he
Yeah.
tweeted left and right nonsense that he didn't know anything about, right? Let's see if he said.
And the same thing happens to PhDs in a number of different fields. I'm going to pick on physicists here because they seem most prone to it. But somebody who becomes an expert in one thing often decides, well, I'm smart. I've got a PhD in physics, and so therefore I know everything, and they will then, expound on many subjects about which they know nothing. It's a hazard.
2022, Prasada Makari, who is the head of the FDA, were co-authors on a paper attacking recommendations for booster shots in teens and young adults, because there was that was the group where you had the most myocarditis,
Yeah.
which, by the way, is much lower than an infection.
Yes.
But then they also get long COVID idiots, because you don't think ahead of time. You don't think, well, maybe there are things we don't know that we should be careful about. No, you just have to blather and get attention. They concluded requiring booster shots in young people would cause more injury than benefit. It did not, because you don't know anything. Neither of you. What else? Prasad's appointment is likely to rattle drug and vaccine makers who depend on predictability of FDA
Yes.
standards and procedures. I think it's fair to say that I'm worried about the standards going forward of vaccines with these guys in charge. I don't think they know what they're doing.
And I'd like to point out a general problem here with predictability, which, people think, oh, Republicans are in charge, they'll be good for business. Business hates chaos.
That's right.
More than anything. You can raise taxes, you can make lots of regulations, you can do all the things that business is supposed to be against, and they'll grumble. But the one thing they absolutely can't stand is not knowing what they're going to need to be spending money on 6 months from now.
Right.
And so when you
Yeah.
throw 100% tariffs on major sources of parts, and you put people in charge of regulatory agencies who are going to go bananas changing regulations, that is horrible, horrible stuff for business. Every business.
Another news item. This is very interesting. The Gates Foundation is going to end in 2045.
Yeah.
It's going to spend all his money. 20 years. So this is an article I got in Gates Foundation website. My new deadline, 20 years to give away virtually all my wealth. So in the first 25 years of the Gates Foundation, they gave away over $100 billion. And they will give over twice that in the next decade, 2 decades. And, he's done a lot for health globally, right? And he's been extremely critical. Oh my gosh, I saw a little quote from him recently about the USAID. He was actually mad about the termination of that program.
Makes sense to me, because
Yeah.
he's spent a lot of he's been in the field, right?
Yeah.
And he knows the populations that are being affected.
So this article you will learn about what he plans to do.
So one of the
next
things that interested me
decades.
about this is I immediately go to the Hughes Foundation, because
Yeah.
Howard Hughes managed to fund a foundation in perpetuity. with-
Almost certainly not what he would have wanted done with his money, but yes.
And so I wonder, proportionally, time adjusted, how much money that takes. And, this article reads like 20 years from now, the money's going to be gone. It will have been spent. Okay. And I wonder about doing that as opposed to endowing something that could last forever. I'm just curious.
I don't know if you can do that, right? That's Hughes had to spend out all its money, right?
You can endow something that'll last forever. I think maybe a reason that Bill Gates is not planning to do that is that would be something whose future he cannot predict, that he would not control.
Good point.
And he would like to push, and this is, when you talk about $1 billion, people often get, it's very hard to conceptualize because it sounds like million.
Yeah.
But just a reminder, $1 billion is 1,000 million. So when we're talking about, billionaires versus millionaires, it's like comparing somebody who's 6 feet tall to somebody who's 6,000 feet tall. All right? Just
Indeed.
picture that, one is kind of above average and the other has his own weather.
Right.
So when $100 billion is a vast sum of money. Yeah, of course, you could start a foundation and have it set up with a board who would be, elected in some fashion, and you'd have some sort of setup and a whole constitutional arrangement. There are many nonprofit foundations that have been around for centuries. that operate like that, and they've got an endowment and they manage it over the years. Yeah, you could do that, but what is that organization going to be doing 100 years from now? It's always possible that
Mhm.
some new directors will come in and decide, we'd really like to be doing something else besides health, and it would drift from his original mission, so he wants to determine how this ends,
So
I
he has
think.
three key aspirations. One, no mom, child, or baby dies of a preventable cause. Two, the next generation grows up in a world without deadly infectious diseases. And three, hundreds of millions of people break free from poverty, putting more countries on a path to prosperity. Now, these are ambitious plans, but it's great. And
Good for you, Bill.
I really
Yeah.
wonder what he thinks of the trend in the U.S. now. He must be really pissed off.
I'm sure he's appalled. Yeah.
Right? The measles alone, right? We were measles free, and now almost 1,000 confirmed cases. It's crazy.
Yeah.
He must be really mad. Anyway, this is very interesting. The only problem I have is he's still going to pour money into polio eradication, where they need to be changing their approach. But all right, that's a story for another time. The next one is, you may have heard the executive order issued by the president, which essentially, it says, purpose, dangerous gain-of-function research has the potential to endanger the lives of American citizens. Of course, it never has in all the years that it's been done, and it's improved the lives, I would argue. If left unrestricted, blah, 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 so they're restricting gain-of-function research and dangerous gain-of-function research, which I think is the first thing here. Who knows what dangerous is? And by corollary, is there safe? What's safe gain-of-function research? What's interesting about this document is they have simply, so the definitions, right? They have simply lifted them from last year's document that the Biden administration put forward, I forgot the committee, NSABB and so forth. So gain-of-function research, dangerous gain-of-function means scientific research on an infectious agent or toxin with the potential to cause disease by enhancing its pathogenicity or increasing its transmissibility. So first of all, I would say nobody knows how to do that. So nobody's going to plan to do that on purpose, because these people think that SARS-CoV-2 was made in a lab. That's what's driving this, and they have no freaking idea. There are a list of 1, 2, 3, 4, 5, 6, 7 things, like enhancing the consequences of a toxin, disrupting immunological responses against a pathogen, conferring resistance, increasing transmissibility, altering the host range or tropism, etc. So that's fine to have this list, but I don't know that anyone could predict what they're doing is actually going to do that. I am on a committee that evaluates these things at Columbia, and we have some work that could potentially fall into this, and it's just not dangerous. It doesn't fall under the dangerous category, in part because you don't know. Now, I am of the ilk that says we've been doing this for many years. We have proper containment regulations in place, and that is actually sufficient to contain it. There's no reason for this because there have been no consequences of gain-of-function research. I think that, if someone were, let's say, modifying HIV to be more virulent, as if you could do that, yeah, okay, that would be a problem, but nobody does that kind of stuff. So anyway, I think this is nonsense, frankly, but it's going to really restrict research.
Yeah, and the way I read this is it's not really, their point is not to decrease risk. That's a cover story. They have simply noticed that there is a category of research that they could easily shut down without having to go through, justifying anything, just by saying we're going to make this safer by banning this whole set of research projects. And then this is something that they can use to continue cutting science, which is the real goal here.
Right.
So several things stood out to me in this. First, in the section 1, there's a sentence here that says, the Biden administration allowed dangerous gain-of-function research within the United States with insufficient levels of oversight. I object to that, among other things, because it's not true,
Yeah.
but also because it's political. Okay? This is the trend of blaming saying we're better than those guys. Okay? That's not the point here. If you're going to regulate research, it doesn't have anything to do with politics. One of the other things they say is that they call it recklessness, if unaddressed, may lead to the proliferation of research on blah, 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 even after COVID-19 revealed the risk of such practices. That's an oblique reference to their notion that SARS-CoV-2 came from a lab, okay? Because that's also untrue, okay? As we've already pointed out, the categories that they list are from old documents, some of which were probably developed during the previous Trump administration. They go all the way back to the H5N1 dust-up in, what was that, 2011? Okay, I think the first set of restrictions came out in 2012, and they haven't changed much since then. The other thing that really struck me about this was, two things, okay? One, they talk about top-down regulation.
Mm-hmm.
Currently, the scientists regulate themselves, okay, more or less, whether you like it or not. Correct me if I'm wrong, Vincent, but it starts at the institutional biosafety committees,
exactly
saying,
right.
is there a problem here? And if there is, then they appeal it to a higher authority, and there are several levels of committees all the way up through the NIH that make the determination as to whether or not this is risky stuff and whether or not it should go forward. The other thing is that this is the typical thing with that's going on now. They carry the money cudgel, okay? So rather than just saying, we're going to evaluate this and restrict it, what they say is we're going to cut off federal funding for anybody who isn't behaving the way we want them to, okay? And it's top down. So those are the big differences to me. It's political, it's top down, and it's all about money. Okay, that's the cudgel.
Okay, and then finally, I don't have a link for this, but apparently it's been 45 years since smallpox was declared eradicated.
Yes, I can tell you. Actually, that's actually interesting. I get different dates now and then for smallpox eradication day. The smallpox was declared eradicated. It's saying 45 years. What year would that make it? 45. I don't know. What I remember
70.
was starting up my laboratory. Finally, after all of this training and stuff,
80.
in 1978. I think 1978 they said this is the last natural case or something like that. I've heard a couple of different dates for the actual eradication, but I remember very shortly after starting up my laboratory, after all this training and stuff, and I'm working on pox viruses, and they declare the disease eradicated. Okay, so what am I going to do now? Well, turned out there was a whole career.
A whole lot more to do on pox viruses.
Yeah, a lot more. The other thing that I wanted to point out is that next week, May 14th, is Smallpox Eradication Day. Because it was on May 14th that Edward Jenner inoculated James Phipps in 1796, okay, and initiated the first experiments with smallpox vaccination.
That was like 1789 or something like that, right?
1796 is my recollection.
Okay. All right, now we have some science for you. Well, we've been talking about science already, but two papers. First is a Cell paper, the recency, and that is not a word I've ever heard. The recency. Oh. Have you, Alan?
Oh, that actually.
It's a word?
It comes up a lot in aviation, as a matter of fact.
Really?
So.
That's in the dictionary. Okay.
Yeah. So how recent something is. The reason it comes up in aviation is because the FAA has detailed regulations about pilots' recurrent training. And there has to be, you have to maintain currency and recency of experience. So it's not enough that you
Okay.
flew 10 times last year. You have to have kept up with it. Anyway,
Go
I can
Go
do a quick
Yeah. Yes,
summary of
please.
this.
Go.
So today's snippet is the recency and geographical origins of bat viruses ancestral to SARS-CoV-2 and SARS-CoV and SARS-CoV-2. It is an uncharacteristically open access paper from Cell. The co-first authors are Jonathan Picard and Spiros Letras, and the 4 co-senior authors are David Robertson, Simon Delacour, Michael Waraby, Joel Wertheim, and Philippe Lemay. If you all are listening, please let me know whether the first author is related to graphic novel writer Harvey Picard. I'm curious. It's an unusual name, and it came up. Anyway, the very international team on this is from the University of Edinburgh, University of California, San Diego and La Jolla, University of Tokyo, University of Glasgow, University of Oxford, University of California, Los Angeles, Scripps Research Institute in La Jolla, Redeemers University in Osun State, Nigeria, Tulane University in New Orleans, University of California, Irvine, University of Hong Kong, China Biodiversity Green Development Foundation in Beijing, Université Libre de Bruxelles in Brussels, KU Leuven in Belgium, Belgium, and the University of Arizona in Tucson. This is another effort to try to narrow down possible origins of SARS-CoV-2 and also SARS-CoV-1. In this case, the researchers mined all of the available viral sequence data on both viruses and the closest closest identified relatives of those viruses within the coronavirus family, and constructed phylogenetic trees to figure out which lineages came from where. A major innovation here is that they took into account the ability of these viruses to recombine and incorporated that when calibrating the molecular clock for estimating when different viral lineages branched off. The results imply that both SARS-CoV-1 and SARS-CoV-2 descended from horseshoe bat coronaviruses, different ones, that circulated between 1 and 6 years before each virus emerged in humans. They also suggest that the immediate ancestors of both viruses had to travel much faster than normal horseshoe bat movements to reach the sites of their human emergences, implicating the wildlife trade in both spillovers. The importance of this work is twofold, I think. First, it is yet another refutation of the conspiracy theories about a lab leak being the source of either SARS coronavirus. And second, it highlights the importance of continuing to study bat viruses and also spillover events into other animals, especially those that are transported for human use.
And to amplify that last point of yours, I would point out that you listed off a whole slug of countries that participated in this, which is necessary. That's the way this has to be done. And I would say point out that funding came from all of those countries as well, including, I presume, a substantial amount from the U.S.
Yes.
So if funding from the U.S. for efforts like this is restricted, these kinds of studies become difficult, if not impossible, to do.
Oh, it's fair to say that all the scientists in charge now, all these contrarians, do not believe it came from nature, and therefore they're not going to fund any research.
Don't refer to the people in charge of science agencies now as scientists, please.
Yeah, they're not.
Unless there's somebody, like, who's the guy at NIH? I think
Taubenberger.
Did he? Was it
Jeffrey
NIH?
Taubenberger, yeah, he's head of NIAID now.
Right, who stepped in to replace what was her name? Who replaced Fauci?
Yeah, I forgot her name.
Anyway, I gather he's just he's a longtime researcher who stepped into the office because nobody else was doing it, and he's a scientist. But anybody appointed by the current administration don't.
Yeah.
Anyway,
so both SARS-1 and SARS-2 are the original source or believed to be bats, including horseshoe bats, Rhinolophus, and likely through transmission through another species. And when the ancestor of these viruses circulated is an important piece of information to get, but it's not easy to do. If you just take the whole genome sequences, which you do then, and compare all these viruses and try to get an idea, of when they were circulating, it's a problem because these viruses recombine extensively. And therefore, when you do these whole genome comparisons without taking recombination into account, you come up with a number that, say, the most common recent ancestor of SARS-CoV-2, using what they call long genomic fragments, gives you an estimate of 30 to 40 years before 2019. But as you'll see in this paper, they take a different approach, and they look at the parts of the genome that have not undergone recombination. They're called non-recombinant regions, or not NRRs. They come up with a very different number. And so that's what they have done in this paper, is to identify the non-recombining regions, which they can do by comparing all the known sarbecovirus sequences. And then for each one, they can establish a molecular clock and estimate when its ancestor was circulating. And then you get a range depending on the different non-recombination sequences that you look at. Right. And so basically, the bottom line, as Alan said, both SARS-1 and SARS-2 likely emerged pretty far from where they were circulating, and they reached the cities where they emerged too quickly based on their calculations here. And so they say they had to have been picked up by a second animal and physically brought to the markets, because that would be the most rapid means. So we're going to go into this.
Well, I mean, the other possibility is that somebody did catch them directly from a bat and then went to one of these places, but that's not consistent with the availability.
That would have to be the same situation in both cases. And in the case of what they are calling, thank you, SARS-CoV-1, we know what the intermediate host was. Okay.
So they begin by taking the 250 full genome sarbecovirus sequences that we have, including SARS-1 and 2, and they use an algorithm to identify recombination breakpoints, and they find 31 putative non-recombination regions for SARS-CoV-1 and 44 for SARS-CoV-2. Okay, so now you have blocks of sequence, and these are about 800 nucleotides, a range of 300 to 3,000, and similar for SARS-CoV-2. So it's both small and large, and then you can work on those to see, to look at the origin.
So the way I think of this is that for any given virus, you could have one of these regions that's really old and another region that's really new. And if you average that out in doing your comparisons, you don't get an accurate picture of where these came from. You have to look at the individual regions, and the algorithms for sorting that out must be every bit as complicated as the algorithms for figuring out the recombination. I mean, this goes over my head, but conceptually I get the idea, and it's really interesting. And I'm blown away by how many recombination hotspots there are. Yes. In each of these, okay? If you've got 31 putative non-recombinant regions, that implies to me 30 recombination hotspots. Or in the case of the other one, SARS-CoV-2, 43, with some non-recombinant regions as small as a couple of hundred nucleotides. So these things are mixing it up like crazy all the time.
Okay, so now they take these non-recombination regions and do a analysis to see what are the closest bat viruses ancestral to both SARS-CoV-1 and SARS-CoV-2. So then they use these various phylogenetic analyses, Bayesian phylogenetic inference. And so the median time of the closest inferred bat ancestor of SARS-CoV-1, of course, it differs in each non-recombination sequences, and it varies by several decades. But the median of these is 1996. I remember SARS-1 emerged in 2003, I believe, right? And then, but the single NRR with the most recent closest inferred ancestor for SARS-CoV-1 had an inferred date of 2001, which is just a year before the emergence of one in 2002. Okay, not 2003. Okay, so that piece was in some virus circulating in 2001. So that's reliable because, this is a recombinant. Well, pieces from all different viruses, which are going to have different ancestral origins. And then for SARS-CoV-2, they do the same thing. The time of the closest inferred bat virus ancestor, again, depending on the NRR, ranged from several years to several decades before 2019, across the 44 different NRRs. But the two NRRs with the most recent time of the closest inferred ancestor had a date of 2014, which is 5 years before introduction into humans. So,
and that you can think of this as these viruses, they're doing all this recombination and they're coming together. And the endpoint that we know came out of this that affected us was the final sequence of SARS-CoV-2 that ended up spilling over into humans.
right.
So if you go a couple of steps back from that virus probably couldn't have gotten into humans. And then you're looking at the most recent events.
Right.
The pieces finally fell into place, and
Exactly.
it, went
So that
through all the holes in the Swiss cheese lined up, and then it came through.
Yeah, so that final piece of the puzzle popped in 5 years before.
5 years before the
and then
spillover.
it went in. Yeah.
So
That's
a
good way.
couple of things interest me about this. That it's the ancestor that they're fingering here is a hypothetical ancestor. Okay, we haven't identified this thing in bats. Okay,
We do not yet have the smoking bat.
right.
Yeah.
But they also point out later that we're not likely to get the
Right.
smoking
We're not gonna
bat
find
because these things continue to evolve. It's probably gone.
Yeah.
Exactly
Unless it
right.
had some selective advantage in bats and
And
enough
hangs
of
out.
it stuck around that it might still be there. But, it's...
Yep. So by this analysis, you can see that the time of the closest inferred ancestor of one is nearer to the date of emergence than for SARS-CoV-2. Okay. And they want to know why that is. And they say one possibility is that we have a large number, more SARS-CoV-1 related viruses than SARS-CoV-2 genomes in bats. So maybe it's that. But if you look at the spike gene, both SARS-1 and 2 have older closest inferred ancestors across the spike gene, and they give you 1963 for 1 and 1944 for 2, just based on the spike gene, which is really interesting. So
If you look at the end, the limitations of the study, I looked through that, and to me, the bottom line there is that the biggest caveat in all this is the sampling.
Yes.
Yes.
Okay, the more sampling you do in the more areas, the better refined a picture you're going to get. Okay, so a lot of these questions may come down to sampling.
In fact, the next section of the paper, they want to look at the influence of sampling on their data. And so basically, they look at the respective recombinant common ancestor, REC-CA, of these two viruses, and they further the genome over time by adding published genome sequences to the reconstruction based on their publication date, right? Because the older viruses could not have pieces of the genome that you find more recently. And so what they find is that as more genomes are published, the SARS-1 REC-CA, this respective recombinant common ancestor, and the two become more similar to 1 and 2 respectively. And so they have a number of different viruses that they cite being similar to that. And by the end of 2022, the REC-CA of SARS-CoV-2 shares 98.8% genome identity with SARS-CoV-2, and the REC of SARS-1, 98.7%. So both unexpectedly exceed the genetic identity of the bat virus genomes that are most genetically similar. So basically what they see is that you add genomes that descend from a more closely related ancestor. Despite them not being the most genetically similar, they underscore that you have to infer these non-recombination reasons in evolutionary analysis. And then basically the greater number of samples of one viruses than two is not solely responsible for the times of closest inferred ancestor because of this analysis that they've just done. But still, we need more samples. But
Yes. Yeah, one of the things that I liked here, though, is by looking at both SARS-1 and SARS-2, SARS-1 I think of as kind of a control here.
yeah,
Because
well,
we do know what the path was. There are a few little gaps, but we know it went from bats into palm civets into people, and in that case, we actually got to the isolating the final step. We are now pretty sure, I think, SARS-CoV-2 went from bats to something. My money's on raccoon dogs to humans, and that's what we're really interested in at this point. But the SARS-CoV-1, the fact that they find, alignment between these two things, it's just a really good parallel to see that everything lines up with what we already know.
Right. Okay, so now, these phylogenies are linked to their hosts' ranges. So they want to know what they call the spatial temporal spread of the two viruses in bats. So they fitted a spatially explicit model to each non-recombination region. In other words, not only what it's related to, but where it was sampled, right? They point out here that it's really hard to look at the deep evolutionary history of RNA viruses that rapidly evolve because of substitution saturation. And what that means is at any one position, if you go back far enough, there's so many changes that have occurred, you don't know what the ancestral state was because it can go back and forth. There are 4 different bases at each position, and they can go and change throughout that time. And so this is called substitution saturation, and they can devise models to account for it. And one of them is called the prisoner of war model. It's a molecular clock that estimates divergence times between viruses, but accounts for these time-dependent evolutionary rates. All right, so they find that substitution saturation primarily affects old more than 50 years old. Okay, that more contemporary viruses, not much of a problem. Okay, so then they go back to the dispersal history, and they look at so they use this prisoner of war phylogeny, and they look at the movement of each non-recombination region across Asia. And basically what they find is that the lineages are spatially structured with similar viruses basically sampled in geographically similar regions with very little back and forth travel across pretty large regions of the study area. So they're pretty constant.
This makes a lot of sense with Rhinolophus bat behavior in biology. These are bats that are kind of homebodies, I guess. They have a home range. They tend to stick to it. When they mate, they may go to another hibernaculum, but they're not long-range migratory bats.
Right. So where is this? Because they say determining where the ancestors circulated, it could help us to do sampling and guide our understanding of how they moved around. And so what they find is they do more phylogeographic analyses to infer this. And the closest inferred ancestors of SARS-1 circulated in Western China, which would be Yunnan, Sichuan, or Guizhou. And for SARS-CoV-2 in Yunnan or northern Laos. And these are where these karst caves are in these areas. And remember, we did a paper out of Laos on the banal viruses, which are very, very closely related to SARS-CoV-2. And so basically, both SARS-1 and 2 emerged into people 1,000 kilometers, over 1,000 from where their closest bat inferred virus ancestors circulating, right? So one emerged in Guangzhou and 2 in Wuhan. And these regions overlapped with the areas where you have the most richness in the horseshoe bat species, which is where these viruses are coming from. The viruses that are most genetically similar to SARS-CoV-1 and 2 both at the genome level and in these non-recombination regions, have been mostly sampled in and near southwest China. So they say the findings are not a result of localized sampling efforts, but they reflect a natural pattern of in the geographic distribution of these viruses. Okay, how about spread? This is very interesting. How fast does SARS-1 and 2 move across Asia? So they calculate a weighted diffusion coefficient. This is very interesting. It measures the diffusivity of viral transmission for the 50 years before the most recently sampled genome in each data set. And here are the numbers, folks. SARS-CoV-1 like viruses spread at 1,666 square kilometers per year, and SARS-CoV-2, 740 square kilometers per year. Now, horseshoe bats, which are the hosts of these viruses, they basically forage 2 to 3 kilometers around their roosts, and they have a diffusion coefficient of 2,000 square kilometers per year. So basically, the viruses are diffusing similar to their hosts, right? The horseshoe bats. That's very cool, I thought.
Yeah, so the virus goes to a neighboring and a neighboring and a neighboring and diffuses across a couple thousand square kilometers. That does not mean that it covers 2,000 linear kilometers.
Right.
It's
Square.
just, square, so it's covering an area.
And also they say, one bat is not going to move all that much. It has a range. But when they roost, different bat species all roost together, so they exchange their viruses, and then another bat can bring it somewhere else, and then the same thing happens. And eventually you can go very long distances, right? Okay. Then they want to know whether the bat virus ancestors of each of these viruses could have spread to where they emerged via dispersal across bat populations. So they determined the minimum distance from the location of the closest inferred bat ancestor to the province of emergence, and they calculate the dispersal velocity, and basically they find that it's unlikely that the lineages descending from the closest inferred viruses reach them solely via dispersal through their bat reservoirs. It happened too quickly for the bats to carry them there. Let's see, what else do we have here? This one I didn't like too much. Yeah, that's basically the data that I wanted to talk about.
I'd be interested. I don't know if you can do this in a meaningful way to see somehow the animal trafficking or human trafficking of animals overlaid on these maps.
Yes.
But my guess is it's just a big smear that humans are trafficking animals all over the place. And so it would be hard to form a correlation, but I don't know because I haven't seen the data.
Yeah, if you could get data on where the farms are for these wild but farmed animals, especially that are the main source for the markets, and where they're hunted in the wild, if those cluster in some areas, and then you could maybe map the routes, but I gather a big problem with this is that a lot of this wildlife trade is, if it's not outright illegal, it's kind of gray and maybe not well documented. So that those would be hard data to find, but I agree that'd be a great thing to look into.
So basically what we have here, these two viruses, SARS-1 and 2, circulated in horseshoe bats hundreds to thousands of kilometers away from where they emerged. And they emerged as recently, and this was circulating 1 to 6 years prior to the emergence. And there wouldn't have been enough time for the bat ancestor to reach the location if you consider the normal bat dispersal routes. By the way, these two viruses and their ancestors have been circulating probably, they say, for thousands of years in these regions. So they're not brand new viruses. And so how did they move so quickly? Well, human-mediated transport of infected animals, like the palm civets. So for SARS-1, the movement of palm civets and raccoon dogs as part of this animal trade is thought to have led to the emergence. And so long-distance transport of those infected animals could account for the speed, right? And then they say the clear evidence that the epicenter of SARS-CoV-2 was at only one of 4 markets in Wuhan that sold wildlife from plausible intermediate host species, infers that the ancestor moved from the surrounding province to an area around or in Yunnan province to Hubei via the wild and farm animal trade. So all the data is consistent with that and not with a lab leak. Lab leak doesn't fit into any of this. And then they also talk about how the bats roosting multiple species, and therefore one species could give a virus to another or variants of a species and then move it longer distances than the bats go. Now let's see. What else can we talk about here? So basically, one thing I forgot to mention. They say at the beginning, SARS-CoV-1, which is currently extinct. So there's no evidence that it's circulating anymore. And there are probably descendants of it in bats, but they're very different now from SARS-1. I think this is humorous in a way because this new NIAID influenza vaccine initiative proposes to have SARS-CoV-1 in the vaccine, in the multivalent vaccine, for which there's no basis at all.
Wow.
Yeah.
You don't need to have SARS-1.
No.
It's extinct. SARS-2, okay, because it's circulating, obviously.
And also SARS-1, because of its transmission mode and its transmissibility, I don't think we really need a vaccine for that. It's not and it's nowhere near as contagious as SARS-CoV-2.
But it just seems to me to underscore that they haven't thought about this.
Yeah, no, of course not.
Anyway, they call this the pieces of a complex puzzle, these mosaic genomes. I like that. That was good. Okay.
And this paper also underscores yet again that we were warned. Not just by everybody in epidemiology and infectious disease, but by nature itself. SARS-1, 2002, we knew this was a danger. That there are viruses out there that can spill over into humans, cause a respiratory disease. We knew they were coronaviruses. We knew what kinds of things caused that. The wildlife trade was a major focus there.
Yep.
And yet, those human systems were still in place 20 years later, almost 20 years later, and you get the big one.
Yeah, we should have been ready. There's no question about it. Got kind of complacent for many reasons.
Ready, and also maybe made some more effort to prevent.
Yeah.
In some respects, we were ready. I think the mRNA vaccine
that's true.
was really ready to go.
And the virology, the knowledge we had about coronavirus spike and how it binds and what protective antibodies look like.
I think the coronavirus vaccine was, if I understand it correctly, was poised basically the way, what's the Center for, or the Collaboration for Pandemic Preparedness? CEPI. I think the mRNA vaccine was essentially poised in the fashion that they would like potential pandemic viruses to be poised.
Sure.
All of the, we knew how to do it. The small animal studies had been done. It was ready to go. All was needed to make the appropriate modifications to fit in this particular spike protein. CEPI must have funded a lot of that stuff. I mean, that was...
I believe they did. Yeah. Yep. All right. Our paper today, which is actually shorter than the snippet, but that's okay. Alan, can you give us a summary?
Yeah. So today's paper is single-dose replicon RNA Sudan virus vaccine uniformly protects female guinea pigs from disease. This is published open access in Nature Communications. The first author is Kyle O'Donnell, and the senior author is Andrea Marzi. The team is from the National Institute of Allergy and Infectious Disease branch in Hamilton, Montana, HDT Bio in Seattle, the Public Health Agency of Canada in Winnipeg, and the University of Manitoba, also in Winnipeg. I guess this is an all North American team. So Sudan virus is a filovirus in the Ebola virus genus. So it's an enveloped virus with a single-strand negative sense RNA genome. An outbreak of it in Uganda in 2022 to 23 killed over 200 people. About half of those, that's about half of the ones who are known to have been infected. So this virus is bad news when it does spill over. There are currently no approved vaccines or treatments for it. And these folks decided there are some things that people are looking at on a preliminary basis. And I think there might even be some that are moving toward clinical trial. But these folks decided to make one using an RNA construct that can replicate after getting into cells. That's in contrast to our non-replicating mRNA-based vaccines that we've used for, say, SARS-CoV-2. Using a replicon, a replicating RNA, has several potential advantages over non-replicating mRNA, including the possibility of giving protective immunity with a single dose, which would be critical in stopping a fast-moving Sudan virus outbreak. To deliver the RNA replicon, they attached it to something called a localizing cationic nanocarrier formulation, or LION. They vaccinated guinea pigs with it, and they found that it elicits a strong antibody response and protects the animals from death, all of the animals from death, when they're challenged with the virus 3 weeks later. And the importance of this is it's a promising preclinical demonstration of a vaccine platform that could be effective in combating not only Sudan virus, but other filoviruses and maybe other viruses in general.
So there are 4 different Ebola viruses. We have Zaire, Sudan, Bundibugyo, and Thai Forest, and Reston, 5. Okay. And most of the outbreaks are Zaire. And we have 2 vaccines against Zaire Ebola virus. We have the VSV based Ebola glycoprotein vaccine, Ervibo, and then there's a 2-dose adenovirus and modified vaccine, Ankara vectored vaccine. And the VSV Ebola doesn't give any cross protection against Sudan virus, of which there are cases, not as many as Zaire, but you heard. But the MVA component of that second vaccine does include the Sudan virus glycoprotein, but the cross protection has never been measured in a clinical setting, so they don't know if it would protect against Sudan at all. You seem like you want to say something, Rich?
No, I'm good.
You're just sniffling. Okay.
I'm just sniffling, yeah.
So you know that there are two kinds of RNA vaccines, right? The ones we all know, the mRNA vaccines that were recently developed, where you have an mRNA in a lipid nanoparticle delivery system. And then you have replicon, self-replicating mRNA vaccines, which have not only the antigen encoded in the mRNA, but they have an RNA polymerase that allows this RNA to replicate and amplify itself in the host. And so for Sudan, their goal is to have a one-shot vaccine that protects against disease. And that's hard to achieve with the mRNA vaccines, they say. So that's why they're investigating these self-amplifying mRNA vaccines. You know, I just have to say that these idiots are so critical of the mRNA vaccine. Did you hear that the Minnesota legislature is considering making mRNA vaccines a weapon of mass destruction?
Oh, God.
Can you imagine this, where there's no evidence that it causes any harm? What a bunch of idiots. So I don't know how these self-replicating vaccines would do. And maybe for Sudan Ebola virus, because Africa's smarter than us in many ways, they say, yeah, protect me against disease. But boy, it never happened here, I think. It's a real problem. By the way, we previously talked about self-replicating mRNA vaccines in TWiV 1115. I think there was such a vaccine licensed in Japan already.
Yeah, I think they mention one in here. One of the many SARS-CoV-2 vaccines, right, was the one developed in India.
Yeah, that's right.
And that one of the COVID-19 vaccines, got emergency use authorization in India after demonstrating robust safety and immunogenicity in over 3,000 participants in a phase 2/3 trial. So this has been deployed, this approach. And when we say, let's just emphasize that when we say self-replicating, this RNA can get into a cell, can make more of itself in that cell, and boost its effective dose in that cell so you get good expression of the protein. It does not have any of the genetic machinery necessary to make a viral particle and get out. So it's still a one-way trip. It's still not going to go anywhere from the person.
Yeah, and it's not going to replicate indefinitely. It's for a period of time.
This is not a live vaccine.
I want to elaborate just a bit and ask a nerdy question. So this vaccine, I mean, it's based on a virus that does this, okay, that has two segments, one of which has all the replication functions and the other of which has the capsid functions. And what they've done is to replace the capsid functions with your antigen of choice. So, it's capitalizing on a virus lifestyle to start with. So,
now,
is this a recombinant viral vaccine or is it an RNA vaccine? Because I've had this question with my, my vaccine group. It's an
RNA vaccine, but it's a recombinant RNA, right? Because you got the antigen and you have an RNA polymerase in there.
Actually, apparently, you can do this in a couple of different ways. You can, there are versions of this, if I understand it correctly, where you can take the genes involved and put them through a cell system that will give you a virus particle out the other end. So it must be a packaging system. So you can deliver that as a virus particle that's non-replicating that helps deliver the RNAs. Or you can deliver the RNA as a nanoparticle. I would say that the first maybe is a recombinant viral vectored vaccine, and the second is an RNA vaccine. This becomes important for me because this group that I work with has templates to describe vaccines that are divided up by categories, one of which is a viral vectored vaccine, the other is an RNA vaccine, so which is it?
See, your basic problem there is you're trying to apply clear categories to biology.
Good point. Yeah. Good point.
Which we do all the time, but it always fails.
Well, for regulatory purposes, sometimes you need to do that, right?
Yeah.
It's hard.
Yes.
So this is the other interesting point about this self-replicating vaccine. You can deliver it in two ways. You can do the traditional lipid nanoparticle, right, where the RNA is inside, and then you inject that, and then, as Arlen said, the RNA replicates inside the cell. But they say that those are quite reactogenic, so you can't increase the dose a lot. It's they say the reactogenicity of these in humans is a challenge. But now we have a different kind of delivery system for these rep RNAs, oil and water emulsion-based cationic nanocarriers. And the one they use here is called Lion. And basically, these decrease the inflammatory responses, so you can give more of a dose. But in this case, the RNA is on the outside of the vehicle, of the nanoparticle. It's absorbed electrostatically to the outside. It's not inside of the particle.
I found this kind of conceptually interesting that I think of something being encapsulated in a nanoparticle would be less immunogenic, just intuitively, because it's, all sealed up in there. Whereas if you stick it on the outside, wouldn't that be more immunogenic? But of course, the RNA is less immunogenic than the nanoparticle, I guess.
This issue of reactogenicity, which is a nice way of saying wipes you out,
yes,
is significant, and I think a lot of people can identify with this. The mRNA vaccines, and I think if I anecdotally, I would say that Moderna, in particular, really wipe people out, and I think that was what I'm seeing here is that's mostly the delivery system, not necessarily the RNA.
Yeah,
and so this is an advance in the technology that addresses that issue, and that's really important to me.
And that's the study in India that someone mentioned, at 3,000.
Yeah,
it's been EU8 in India, and it was safe. Not bad reactogenicity. The other point about Lion is you don't need cells to make them, right? You just assemble these in a test tube. And so these vectored, if you want to make the virus, recombinant virus, as Rich said, you have to use cell cultures. But here you just put chemicals together. So basically what they are doing here is evaluating these lion-based Sudan Ebola virus self-replicating vaccines in a guinea pig model of disease. They're looking for protection against disease. Okay? That's the way you do it. And they want to do one dose. So basically they immunize, they challenge 3 weeks later, and they find one dose of the lion Sudan virus vaccine protects 100% of the guinea pigs against disease. Now, guinea pigs are not humans, of course, so we'll have to be tested in people, but this is good. It allows you to go forward.
So, the sense I got from them about the importance of the one dose thing is that these, historically at least, and probably going into the future, these vaccines are deployed in defense of an outbreak. Okay,
Yes.
It's not like you go through a whole population and immunize everybody. So you got time to do a vaccine and a boost, and everybody's okay. Because the frequency and prevalence of these outbreaks is probably not big enough necessarily to justify that, except in certain populations, I suppose. But what they do is they do this ring vaccination thing where they identify where the index cases are and then vaccinate outwards from that. And time is a big deal. So if you've got a one-dose vaccine and you don't have to, you can be immune in 2 weeks instead of a month or 6 weeks. That's and you only have to hit anybody once. You don't have to follow up. That's a big deal.
Yeah, and especially in the places where these spillovers are occurring, not having to follow up and track people down for a second dose is a big help.
Mm-hmm.
So they construct these rep RNAs. They first take the wild-type Sudan glycoprotein, where they codon optimize it for guinea pigs, and they find it doesn't matter in terms of inducing antibody responses. So they stick with the wild-type sequence. And then they vaccinate groups of guinea pigs, 6 each, and then they challenged them 21 days later. So they have a lot of different viruses or preparations that they use. They have the rep RNA lion construct with either the Sudan glycoprotein or a combination of Sudan and Zaire Ebola virus glycoproteins, or the Zaire glycoprotein alone. Okay? And then they also use the VSV Sudan vaccine that was developed that hasn't been licensed yet. Okay? So not the VSV Ebola Zaire, but VSV Sudan. And this has been shown to have protective efficacy in non-human primates, so that's a positive control. Right? 6 of the 6 guinea pigs that get lion, Sudan, or the lion combination, which includes Sudan and Zaire, survived challenge. And the controls that got nothing all died.
You don't want to be the control guinea pig in this experiment.
Now, 3 of 6 guinea pigs that got the lion Ebola Zaire survived infection. So that shows some cross-reactivity there, at least in this system. It's interesting. 5 of 6 guinea pigs that got VSV Sudan survived challenge. Only one died. So the rep was better, I guess, although that's a small number.
Yeah.
And all the controls died by 9 days post-challenge. So in terms of protection against disease, this lion Sudan works really well.
Yeah.
In guinea pigs. Single dose in guinea pigs.
And did you mention the lion Ebola virus vaccine in these? Half of them died?
Yeah, he mentioned that.
Yeah, okay.
Yeah.
Got that.
So I'm looking at the graphs of weight loss and temperature.
Yeah.
I mean, the animals that were immunized with the good stuff didn't even spike a fever. They just shrugged it right off.
Yep. So looks good. What about replication? Let's take a look at replication in these animals. Virus in the blood, liver, and spleen, 6 days post-challenge. So if you got lion, Sudan, or lion combination, or VSV Sudan, they had much less RNA and infectious virus in the blood. So they actually look for infectious virus, which is great.
Yes.
This is really good. they can't find any RNA and infectious virus in liver and spleen samples of animals that got it the Sudan or combination vaccines, and they go on. In most cases, the virology parallels the clinical observations. They look at some pathology now. They take some sections of different tissues. Guinea pigs that got lioness Sudan, lion combination, or VSV Sudan had no or mild pathology in the spleen and liver, and limited to no antigen-specific immunoreactivity, and in the control groups, they had lots of damage in the tissue, lots of degeneration and necrosis, infiltration, vacuolar degradation, etc. So at the level of pathology, the lion vaccine is quite good as well. They note that we can't do T-cell studies in guinea pigs because we don't have any reagents for doing that. So they tried to find a correlative protection. They look at different days. They have -7 days a week before challenge, and 6 days after challenge, and 28 days after challenge. And they look for antibodies either by binding or by neutralization. And the interesting thing is that protection is mediated not only by neutralization, but also by FC-mediated functions. I'm summarizing a lot of data here, but I don't think we need to go into detail. So FC-mediated functions mean an antibody, the FC portion can bind to a receptor on a cell, and then that cell can eliminate, say, virus-infected cells. And so, and also complement-mediated functions. These serum complement proteins can utilize antibodies in a similar way. And so, depending on the vaccine, actually, so for example, the lion Sudan stimulates a neutralizing antibody profile, but also stimulates phagocytosis by these FC-mediated functions, whereas the VSV vaccine induces a neutralizing and complement-mediated protection. So it's very interesting how the vector can make a difference in the kind of immune responses that you get. So that's it. That's the data that one dose of a self-replicating RNA vaccine delivered in these lion particles on the surface can protect in guinea pigs against challenge. So that bodes well for, I guess, further development. I guess they'd go into non-human primates next.
Yeah.
Yeah, I
Well,
really
maybe some other animals. They mentioned ferrets as one possibility. Something cheaper before you go into non-human primates.
Did you know that ferrets are not people?
They're not people. No, but neither are guinea pigs.
Yes, they aren't.
I really like this. I like the lion carrier being less reactogenic, and I like the idea that you can, because it's self-replicating, you can start with a smaller dose to start with, and you don't have to necessarily do the boost. Though it'll be interesting when we get around to human trials with something like this. I can't imagine that they won't do trials that do a prime and a boost versus just the prime. Same. We went through this with SARS-CoV-2, right?
Yeah.
Yeah. And also look at longevity of the response.
Yeah. Though, actually, I would prefer not to have another situation where you could actually do a placebo-controlled trial. Okay, because that implies that you would be dealing with a new pathogen, and you would be able to find people that had never been exposed to it that would be appropriate for the placebo.
Oh, are you referring to the fact that they want to have Novavax do a placebo-controlled trial for its variant booster?
First of all,
it's unethical,
correct me
right?
if I'm wrong here. The notion that there have not been placebo-controlled trials for vaccines is false.
Yeah.
When vaccines first are tested, okay, they typically do a placebo-controlled trial. In the case in point, SARS-CoV-2.
Yeah.
And it's easiest to do if you have a population that's not seen the pathogen, so that the controls will actually get the disease.
Yeah.
Okay. But what you don't have to do that indefinitely. If you've established the efficacy of a vaccine, you don't have to do a placebo-controlled trial every time. Plus, it's not necessarily practical when you have a population that's been continuously exposed to this.
Yeah, and it's not ethical if you have a standard of care, you can't withhold it.
Yeah.
They also said that the placebo should always be saline, which is wrong.
No.
It's totally wrong. These guys don't know what they're talking about. So anyway, yeah, we're in for tough times, folks.
Yeah.
All right, let's do some email. Alan, can you take that first one?
Yes, let me get the right window here. Okay, so Rachel writes, "Hello, TWiV team. Enjoyed this last episode, but thought I would just put out a little correction. Currently, almost all, if not all, veterinary schools and even veterinary technician schools in the U.S. require rabies vaccination and often subsequent titer checks. Prior to 2022, we all received a 3-dose series, but the guidelines were updated in 2022 and changed to a 2-dose protocol. Most of us did spike a high fever with some malaise, especially after dose 2, which lasted 1 to 3 days depending on the student. Some schools even make funny t-shirts with phrases like, might bite, but it's okay, I'm rabies vaccinated, etc. At the annual AVMA convention, American Veterinary Medical Association, they even host a booth for vet staff to get their blood drawn for titers at the event. Just an interesting fact about rabies vaccination in the U.S. Thanks to you and your team for continuing to spread great information during these times." Well, thank you, Rachel. And that was, I don't remember what episode that was regarding.
So last time Angela was on, she was saying that they don't need to get rabies vaccination.
Yeah, I think you weren't here, Alan.
Okay.
And I thought that they always had to, but we have a vet now who says... Well,
apparently that is current practice.
Well, and that's in the U.S.?
Right.
Okay, yeah, Angela's in Canada?
Yeah, it could be different, right?
Right. And Rachel is apparently now is a DVM, and she's a biomedical and bioscience PhD student at Cornell.
That's interesting. That's what Angela is. She's a DVM getting a PhD.
Getting a PhD.
That's standard stuff. If you
Seems
want an academic job in veterinary medicine, your DVM isn't good enough. You got to have a PhD on top of it.
Cool. Very cool. Rich, can you take the next one?
Eileen writes, "Hi all. I love TWiV and all of you. I discovered TWiV in fall 2020 and haven't missed an episode. Immediately ran back to January 2020, plus started back listening to day one, and am currently to May 2020. You got a long haul, man. I am a registered nurse, mom of 2, and a rabid pro-vaxxer, no pun intended, as upset as you all are about our current situation. Episode 1213, towards the end, you mentioned Oprah Winfrey. She was mentioned in the context of a Malcolm Gladwell episode as a person who really is penetrating in her interviews. I have never watched her program, but I'm familiar with her history, etc., and coincidentally had just been wondering earlier today if she has ever expressed any regrets regarding her promotion of both Dr. Phil and Dr. Oz. I don't know the answer to that. Do you guys?
No, I don't.
know.
She says, I wouldn't be able to sleep at night. Thank you all for all you do and the sense of comfort and companionship you have provided me and countless others, I'm sure, over the years. I loved and missed Miss Dixon, and my heart goes out to all of you for the loss of such a kind and wise friend and colleague. Sincerely, Eileen. Thank you, Eileen.
Thank you.
That's very nice.
Yeah, I think without her, Dr. Oz would not be head of whatever Medicare thing he is today, because he got very prominent because of her. But he would promote crap on her program.
Yeah.
Right? But someone like that who's very successful doesn't usually apologize for stuff like that, right?
Or, I mean, even if she did, it would be too late because damage
April
is done.
writes, "Dear TWiV team, it's 47F, light rain showers, and wind at 17 miles per hour. Rich was talking about motivation. I agree the main driver in our current predicament is greed, but I'm going to give my take on what got us in this place. Racism and misogyny. people could not vote for either a person of color or a woman or both. So they didn't vote or voted for what we got. Regretfully, April. Right on, April.
Right on, April. And
Yeah.
I would point out that let's continue our education here. That the three poisons, greed we picked as the big one, but the three poisons are greed, hatred, and delusion. Okay, and I think that you can put racism and misogyny in a combination of both hatred and delusion. So it still fits the three poisons. I love the three poisons because they kind of cover the whole thing.
Very explanatory, yes.
I just cannot understand what would make a person not vote for a person of color or a woman. We're all human beings. Get over this nonsense, folks. Unbelievable. But that's the world. It's not just the U.S. I mean, the whole world is like this, right? Oh, my gosh.
There's something literally in our DNA that does this. And they're probably evolutionary biologists or psychobiologists who can help explain this. There's going to be in our evolutionary history some sort of fitness advantage to being a racist, right?
There's the simplest I can think of is we are tribal apes.
Yeah.
And we know our own tribe, and traditionally this would be mostly relatives who are around you of some variety, who would probably look a lot like you. And when you see people who look very different from your own tribe, they are potential threats.
Yeah.
Now, of course, in the modern world, we can form tribes out of people who look very different from us, and that can then become our group. But because of long-standing practices, we often have difficulty forming those more diverse groups because we live in neighborhoods that all look like us, and because we've followed, this same type of pattern. And so, yeah, I think there is a fundamental aspect of this, but it can be overcome without a whole lot of effort.
Yeah, well, to me, the next step in human evolution is a conscious development of consciousness and, if you like, spirituality to the point where we can consciously counteract these hardwired prejudices and, bad things, wanting to kill people.
George Carlin proposed an alternate, very colorful solution to the problem that I can't quote it verbatim because it's George Carlin, but basically it was everybody have sex with everybody else until we're all the same color.
Oh, good idea.
Was the George Carlin proposal, which would be an alternate way of doing it.
Yeah, that's a good idea.
Alan, we're back to you.
Sure. So we're up to April?
Mary.
Oh, that's right. Mary writes, Trump tracker currently at 430 billion. Dear TWiV team, thank you for all you do. This morning's Seattle Times carries an article about an attempt by House and Senate appropriations leaders to capture the big picture of financial damage to federal programs from Trump 2. Skip to the tracker section on Department of Health and Human Services to see the extent of the damage to programs you and your followers care about. The tracker has limitations, including that it is only a 100th day snapshot of congressionally authorized funding blocked by Trump, but I liked the attempt to document overall damage done by the felon-in-chief. Warm regards from Mary, and she sends the links to the article and the tracker.
I'm surprised that HHS even has this up.
Yeah.
No.
Well, I guess it's the Democrats.
It's House Democrats.
Yeah, House Democrats
are hosting this.
Felon-in-chief. It's true.
Yep.
Rich.
Dagfin writes, I salute you, the TWiV team, and applaud your courageous efforts to continue excellent science communication in this chaotic time under the Trump administration. While listening to TWiV 1213, I started reflecting on the problem of handling political issues in TWiV. During Dr. Conant's remark towards the end of the episode, it struck me that keeping a science podcast free from politics is a privilege and a confirmation that one is navigating in a political system that embraces science and reproducible knowledge. The increasing amount of political remarks and issues in TWiV recently is a clear indication that this privilege is under attack or may even be obsolete. I do not want TWiV to be political, but I think you have no choice. By all means, keep communicating science. That's why we come to you. But in light of the indecent attacks on the entire American science community, scientists have to speak up. Balancing this towards the purpose of TWiV is, of course, a difficult exercise, but you cannot keep silent. I have tried my best to fight misinterpretations and outright stupidity in my own country, but it is indeed a fight against windmills. But I'll never give up. Politicians want clear answers with 2 lines underscoring a correct answer. We cannot provide that, which is why I was made unpleasantly aware of when I was asked to run for a seat in my city council. Luckily, I wasn't enough of a political talent to acquire the seat, but I learned that I cannot have a position in which I have to make decisions in cases where I need to conclude based on conflicting data in subjects that I only partly understand or do not understand at all. As we scientists need to explain these aspects to people, we as scientists need to explain these aspects to people and politicians. TWiV is an excellent medium to do so. Keep it discreet, but please do not ignore the political aspect of the current conditions. It is, however sad one may think, more necessary now than ever. Keep up the excellent work. Best regards, Dagfin. Thank you, Dagfin, for that validation. I've been thinking about this recently. This need, in quotes, to present things as black and white in a political realm is really too bad. There's no room for nuance. And that's how we get into, because scientists by training won't be absolute about anything. They'll say, the data supports the hypothesis that, the minute you say that, everybody just turns away. Nature and the things we deal with is not black and white. It's all gray. As a population and as leaders, we have to learn how to deal with the grayness, but that's not going to happen anytime soon.
Bob writes, several years ago, I was a frequent listener to Joe Rogan, among many other podcasts to which I subscribed. I have found several other podcasts in what I will call spin-offs from Rogan. One was Lex Friedman, and I listened to him for a while when he first got started with his own podcast. At some point, Vincent was on one of those 2. I think it was Friedman. That's right. And I enjoyed listening to an actual virologist and soon found TWiV by following Vincent and have been listening ever since. I'm a nuclear power plant systems engineer by training and vocation, 45 years worth. So I only understand about 50% of what you discuss. However, I understand science and scientific method and know when I'm listening to those who know and when I'm listening to one who is making things up for an agenda. Thank you, TWiV, for being the former. Oh, Bob, I wish more people were like that. My suggestion, please get out to other podcasts if you can and show off the truth. It could be science, economics, stories, as in Gladwell, but somewhere thinking people listen. Let them know there are real scientists doing real work on viruses, vaccines, etc. Maybe you will only get to a few as you got to me, but it can only help us fight this terrible attack on science and the health of the world we are seeing. Thanks for all the information and great discussions. Bob is in Wilmington, North Carolina, currently 76F and mostly cloudy. Yeah, I haven't been on another podcast in a long time.
Vincent, right, Malcolm, are you listening?
Okay. You should have Vincent on.
You need to get Vincent on your podcast, okay? Really?
Yeah.
I don't even know that. Maybe Lex Friedman was the last other podcast. During COVID, I was on a bunch of others, including Jillian Michaels, by the way, the exercise lady. Okay?
Oh.
I was on her podcast, and she was very into vaccines, and now she's made us 360. Now she thinks vaccines are all bad.
I don't know. Would be a
180. I remember that. Yeah.
180, sorry. 180.
If she did a 360, she'd still be back on board.
Yeah, I'm sorry about that. Let's do another round.
Sure.
Alan.
Lisa writes, hello, thank you once again for the informative content and commitment to providing facts to the public. I do have a small correction regarding Angela's comments in episode 1213 regarding human rabies vaccination. Oh, wait, no,
It's a new one.
It's new. We have confirmation of the previous email.
Yes, okay. In North American veterinary schools, rabies... I thought I had scrolled back up by accident. In North American veterinary schools, rabies vaccination is standard for all students and generally mandatory. Veterinarians will monitor titers throughout their career, receiving boosters if warranted. I am based in the U.S., but I consulted with several Canadian veterinarians who assured me that this was also standard throughout Canada. Informal polling, but I received responses from dozens of graduates of veterinary schools across Canada with graduation dates spanning the past 30 years. Additionally, I am not sure what vaccine Angela was talking about, but the modern rabies vaccinations typically only cause local soreness and short-lived malaise, similar to a flu or tetanus vaccination. Pre-exposure prophylactic vaccinations are given as a 3-shot series. Right, so we have slightly conflicting information here, but basically confirmation.
Close enough.
Confirmation. And a tangential reminder, but post-exposure vaccinations are the only effective treatment for exposure to rabies. They should always be offered after a potential exposure. Rabies remains endemic in wildlife in both Canada and the U.S., with spillover into domestic animals. Thank you so much. Lisa is a DVM in Eugene, Oregon. Thank you, Lisa.
Okay. Good to know. Rich.
Petra writes, hello, wonderful TWiV team. First, I want to thank you and commend you for your rigor around using correct terminology. In grad school, the difference between a mutation and an amino acid substitution was seared into my brain by the amazing faculty in my training program at UW-Madison. I appreciate that you guys insist on the correct wording as well. I listened to your discussion about rabies vaccines and wanted to share my experience. I was exposed to a bat and needed rabies prophylaxis and vaccination. The prophylaxis, pooled human anti-rabies immunoglobulin, was 3 intramuscular injections in the thigh. Because it's a large volume, they split it up. I had no flu-like symptoms from the vaccine series, just a sore arm. I was a postdoc at the University of Minnesota at the time, and I was able to easily get treated at the outpatient clinic at the University Hospital. Unfortunately, I was unable to donate blood for a long time after that because the IG was a human blood product. Okay. On the bright side, it was easy for me to ensure that I didn't get rabies. Thanks for the great show. Petra from Minneapolis. Great. Cool.
I'm so glad you agree with the definition of mutation, because many of my colleagues push back. It doesn't matter. It doesn't matter. No, it does matter.
Yeah, I wonder who the professors were at UW who drilled that into her. I mean, the first person that comes to mind is Ann Palmenberg, right? She
Palmenberg, Roland Ruckert. Who was the herpes guy there? EBV. He was very good. Can't remember. Okay, my turn. The last one. Chris writes, Dear microbial mobsters, the reason I love your YouTube podcast is because I get to be a fly on your wall as you discuss the science. It is my joy to listen in on your conversational style without guile or agenda other than to learn and share scientific research. You are a safe space and always improve my well-being. So thanks for that. No kidding. I think we have an agenda. We promote science, right?
Yes.
You are not trying to convince me of your worldview, just sharing the richness of science. Neither do you pitch advertising endlessly, nor blather on and on to lengthen the podcast, maximizing monetization. We don't monetize.
We blather on and on for other reasons. It's not about money.
Being a loyal fly is a privilege, giving me entry into science unseen elsewhere. Without you, I would be entirely ignorant. A horrible thought. You generously interrupt your reviews to clarify meanings and subtleties so we lay members of your audience understand, which is much appreciated. One request, please share more screens so I may see the graphs and charts as you discuss the papers. I am a visual learner and find your reviews enhanced with these slides when you pop them up. I often hit paywalls trying to find them. You sometimes say the paper is best presented in slide A, which I'd like to see too, please, and thanks. Here on TWiV, we try to avoid calling out figures. because of this, right?
Yeah.
We try and just summarize the figure.
Yeah. Because many of our listeners are listeners only. They're driving or they're walking their dog or something, and they're not watching.
But I
on
know
YouTube.
that in some of the other podcasts, they call out figures. I don't like it, but they don't listen to me. Okay. I don't know what to do. I say, please, folks, don't call out figures. But I think it's ingrained in them because they give talks and this figure. Okay. It is tragic. The U.S. government is anti-science. The cognitive dissonance is seismic. Keep fighting lest civilization go dark. Just a thankful fly, Chris. Toronto at 15C, 69F, cloudy 2 days ago. The trees bloomed all at once. Poof! Big rain, longer sunny day following, and bam, it's spring. Thank you, Chris, and all the letter writers.
So,
hearing.
Chris gets it. I mean, your
does.
original vision for this, Vincent, was everybody should be flies on the wall of our discussions that we have behind the scenes, right?
That's right.
And I'm glad that works.
Still doing it, yeah. It's cool. I must say, people support us. It's really encouraging. People like a small minority of people like what we do. We just hit 137 subscribers on YouTube. We get about 600-700 a month. So figure when are we going to reach 1 million, Alan? Long time.
I don't know. I don't do predictions.
Well, we can just do a rate calculation.
Yeah.
1 million divided by 1,000. That would be easy.
But past results do not necessarily guarantee future performance.
Oh, God. I'm going to just do a calculation. 1 million divided by, well, I could do it by 10 to the 6th over 10 to the 3 is 1,000 months. It's going to take longer than I'm around, folks. All right. Rich, let's do some picks of the week. Rich, what do you have for us?
I'm a little lighter than recently today. My pick is Andor Season 2. Okay, so this is in the Star Wars family of thingies. This is a TV series. So you can find it on Disney Plus, and at this point, nowhere else. As history, you will recall that the original Star Wars film, which is now Star Wars A New Hope, filmed in 1977. Can you believe that? It's almost 50 years ago. Had to do with R2-D2 had plans for the Death Star concealed in him that had been put there by Princess Leia. And Luke Skywalker and Han Solo and Obi-Wan Kenobi discovered this and managed to use those plans to identify a weak spot in the construction of the Death Star and then wipe it out. Then there was a prequel made in 2016, 40 years later, called Rogue One. And that's the story of how they got the plans to the Death Star and planted them in R2-D2. One of the principals in that story was a guy named Cassian Andor. And Andor, the 2 seasons together, a total of 24 episodes, is the backstory on Cassian Andor, who started out just as a crook and over time became sympathetic with the rebellion. Actually, the first season which I rewatched before watching season 2, was his how he was converted from crook to rebel. Okay, and then the second season is basically how he advances through the rebel operation to become a leader and leads up to Rogue One, which I'm going to have to watch when I'm done with this. I've already seen Star Wars too many times to watch that again. So I haven't even watched all 12 episodes of season 2 because they're dropping them 3 a week, and I binge them when they drop, and the last 3 haven't dropped yet. I'm biting my nails till next Tuesday. It's very good. It's really well done.
Didn't know about this.
Oh, it's very well done. This is one of the best Star Wars series out there.
I don't even remember who Andor was. I did see Rogue
One, yeah. Yeah, there were
But
2
who's
characters, a
Andor?
woman named Jin and her sidekick, who's basically an officer in the rebel forces, named Cassian Andor, all played by the same guy. His name is Diego Luna, who is very good, really good. So this is really well done.
Oh,
Cool.
I see his picture there. Okay. Alan, what do you have for us?
So my pick is a bit of an update on an older study. So this is let's see, it is open access. It's a Nature paper, Genomic and Genetic Insights into Mendel's P genes. This is a genome sequence analysis that finally answers a few questions that were left open by Mendel, Gregor Mendel, who did the first genetic experiments, and everybody learned in high school biology with the pea plant flower color and doing the Punnett squares and figuring out what color the flowers are going to be in the next generation. And this is just, I think it's really cool that people are still looking at those questions that remained and finding answers to them. And I'm also linking to an article by Eric Stokstad published in Science. It's a news article covering this same study. So go check it out.
So Mendel did his experiments in between 1856 and 1863. So yeah, this is an update on an older study.
Yes.
Using some 28,000 plants. The guy was a genius.
Yeah.
I mean, it's just, those studies are just amazing. How he was one of the first guys to use math, okay,
yes,
to deal with biology. And he did all this stuff and came up with the conclusion that we were diploid.
Yes.
Okay, which is just fascinates me. And I also, when I used to teach this stuff, I used to try and put myself in his shoes, okay, where you take, I don't know exactly what they are, but they talk about two different colors of flower, say a purple and a white, and you cross them, okay, and the next generation is all purple. And then you cross those guys, the whites have disappeared. And then you cross those guys and holy cow, the whites are back. Where did they go?
Where did they come from?
You know, and where did they come from? Brilliant.
Yeah.
Okay. Alan, what do you have for us?
That's what I just gave
That's
you.
what he just gave
Oh,
us.
I just heard Rich.
Yeah, I was
talking.
I was rapping on.
He was riffing on my
I was
pick,
riffing on it.
which is absolutely fine.
Okay.
I had kind of the same reaction when I saw this. I was like, oh, wow.
I just sent the article to Nels. I want to do it on a TWiV.
Yeah,
It's
that
very
would be
cool.
good.
All right, my pick is a Nature article, which is paywalled, but you can read the abstract, and that's all you need to do. 2023 summer warmth unparalleled over the past 2,000 years. Maybe you just need the headline, folks.
Yeah. Hot enough yet?
2023 has been reported as the hottest year on record, but I'm going to read the abstract. But contextualizing recent anthropogenic warming against past natural variability is challenging because of the sparse meteorological records from the 19th century, and they tend to overestimate temperatures. Okay, that's understandable. Here, we combine observed and reconstructed June-August surface air temperatures to show that 2023 was the warmest northern hemisphere extratropical summer over the past 2,000 years, exceeding the 95% confidence range of natural climate variability by more than 0.5 degrees Celsius. Comparison of the 2023 June-August warming against the coldest reconstructed summer in CE 536 shows a maximum range of pre-Anthropocene to 2023 temperatures of 3.93 C. Although 2023 is consistent with the greenhouse's gas-induced warming trend, that is amplified by an unfolding El Nino. This emphasizes urgency to implement international agreements for carbon emission reduction. All right, this is science. These are the data, folks. They're not fake. They're real. Global warming is real. And if you make up data to say it's not, you're lying and you're disgusting. You're a disgusting human being.
This, to me, everything, certainly in my world, everything is perceived in the context of this issue. Okay? It drives me nuts that with this going on, we're trashing the planet. Not only with respect to climate change, but in a whole bunch of other ways as well. And you know, when the planet's gone, you're not going to be able to be killing each other. Okay, so you're wasting your time killing each other.
Yeah.
You know, this stuff is on the top of my mind, and the vast majority of people aren't even thinking about it. They're fighting with each other over really made up ideological differences, okay, and that kind of stuff. It makes no sense at all.
Well, you know, it involves regulation, which doesn't fit.
Oh yeah, it's all about money,
administration.
and certainly in our situation, and this is something that people don't talk about enough. The false contrary narratives about climate change are all because it goes back to Al Gore's thing. What was out 20, 30 years ago? An inconvenient truth. The inconvenient truth is that if you want to address this, some of the biggest money-making industries in the country are going to have to make some serious changes and even go under. And that's where we're going eventually, whether you like it or not. But people are short-sighted, you know. They say, "Oh, it's okay. Well, you know, as long as I can live out my life in these circumstances, to heck with people down the road." No, it's about money. It's about money.
Greed.
Yeah, there you go.
What are the three poisons? Greed,
hatred, and delusion.
Hatred and delusion.
Okay.
Okay, and
Delusion is
remember,
part of it too.
the opposite. Delusion is a little hard to understand unless you consider the opposite, which is wisdom. Okay, so the opposites are to me as important as the three poisons. They're generosity, love, and wisdom. Okay, and wisdom is, you know, knowing the truth of what's going on. I'm in correspondence with a TWiV listener about what truth is. You know, actually, you know who asked that question?
1 +
What
1
is truth?
That's truth. That's truth.
A lot of people.
Pontius Pilate.
Yes.
Okay, in Jesus' conviction, okay, was confronted with trying to figure out what the truth of the situation was. Okay, and he says, "What is truth?"
Well, he's BSing it. Because in that particular situation,
But
it's very clear.
he was not the first to ask that question,
Right.
though.
No.
And I mean, we are not privy to probably capital T truth,
Right.
but what we can get, the closest thing, is transsubjective truth, which you get with science. Truth that's true whether you believe it or not.
Yeah, this is the nature of this conversation I had, because I made the comment that some truths are subjective,
Yes.
and that rightfully was there was exception taken to that. Capital T truth is not subjective. Capital T truth is science.
Or the closest we can get to capital T truth.
We have a listener pick, very interesting, from Brian. He sends an archive.org article. A large language model benchmark that measures the capability to troubleshoot complex virology laboratory protocols. So this is an article which it's a large language model that measures the capability to troubleshoot complex virology laboratory protocols. I guess he just said that. That's the first sentence. These results, and this is a quote, these results indicate that frontier models have been able to match or exceed the ability of human experts to provide practical troubleshooting assistance in virology for over a year with the release of Gemini 1.5 Pro in February 2025, and that the disparity between humans and models is widening. Interesting times we live in, writes Brian.
So I don't get it. Does this mean that when my cells are no longer growing, that I can feed everything I know into this AI, and they'll tell me what's going on?
Yeah, so it consists of 322 multiple part questions covering fundamental knowledge to work in a virology laboratory.
I would be interested to play with that. It's very interesting.
Yeah. This is very interesting. So recently on Twin, we did a paper. So Google just released another basically AI program that helps you write grants, basically. But it identifies new problems in a field. And so there's a Nature article on it where these investigators fed it the data that they had, and it came up with some new hypotheses. So it designs experiments, basically. It came up with new hypotheses that the authors had thought of themselves but hadn't yet gotten around to. The program did it, and they did the experiments, and they got answers. And so I forgot what it's called. Scientist like 2.0 or something like that. It's amazing what it does. Holy cow. And it looks bona fide. It's all built on information you give it, obviously. These are language learning models, large language learning models. But, man, I don't know what they're going to do with it. They're going to probably sell it to companies and stuff like that. But interesting times we're in. All right, that's TWiV 1217, and they will never replace podcasters.
No.
No.
Because we just veer off into all kinds of things because of our brains.
AI wouldn't tolerate this at all.
No.
Microbe.tv/TWiV is where you can find the show notes. You can send questions, comments, and pics to TWiV@microbe.tv. And again, we'd love your financial support. We love to do these programs. We learn a lot. We hope you learn as well. Microbe.tv/contribute. Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Condit is an emeritus professor at the University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at microbe.tv or on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.